Hey everyone, welcome to our webinar. My name is Kasia Proctor and I am the Director of Marketing Communications here at Biolog. I will be your moderator for this webinar. Our webinar today is titled Learn from the Experts Why TruPRAS Media is the Key to Consistently Growing Anaerobes. Let me introduce our speakers. Steve Cox is VP of Operations at our Morgan Hill location. Steve has been working with anaerobes since he was a child, starting when his dad, Mike Cox, the founder of Anaerobe Systems and the inventor of the PRAS media, would bring him and his sister to work on the weekends and summer breaks. Steve grew up working through different departments, from media production to automation engineering, and after earning his degree in industrial engineering, he designed and built new and improved gloveless anaerobic chambers. The decades of experience working with customers in their labs and improving anaerobic media at the company have made Steve a true expert for all things anaerobic. And Dina Fanon, VP of our marketing here at Biolog. Dina received her PhD in biochemistry from Stanford University, studying molecular motor proteins and their role in embryonic development. She had worked in industrial biotechnology, engineering enzymes for a variety of applications. Prior to joining Biolog, she has spent many years developing and commercializing life science tools to help accelerate research. Thank you both for your time. Dina, take it away.
Thanks, Kasia. All right, so the first part is about TruPRAS media and really why that's key to getting consistent growth of anaerobes. And so I'm assuming that most people who are tuning in today are doing so because they probably already work with anaerobes and maybe they're looking for tips or better tools to help them. But I do think it's super interesting to take a big step back and think about our planet a long time ago before there was any oxygen in the air at all. And these anaerobic organisms could really thrive. Now, of course, the world we live in has about 21% oxygen in the air. And so anaerobes have been sort of forced to occupy hidden environments that are still oxygen free. Despite that, though, they are still such an important part of pretty much all of our ecosystems, whether that's in our body or in the body of animals or in the soil or the water. So really, if you're trying to do research or clinical work on these organisms, many of which are, play important roles in infections, it's really critical to be able to maintain the right conditions to be able to reliably culture them and grow them. So Steve, you have such a long history, a family history, of developing tools to make it possible to cultivate these types of organisms. Can you just talk a little bit more about how you got started in this area?
Thanks, Dina. Well, it actually started with my dad, Mike Cox, well before I was born, who really got into this industry. So he started out working as a microbiologist in a clinical diagnostic lab, and him and his colleagues were really struggling to isolate and culture organisms from infections that they knew and suspected were anaerobic bacteria, but they did not have the tools, the media, the anaerobic chambers to be successful in culturing them. So they would just report out mixed anaerobic infection and did not lead to great prescriptions for antibiotics and treatments. So he started his company himself to really bring the best anaerobic culture media and new innovations in anaerobic chambers to microbiologists to not only make his work more successful, but to help everyone else out. So he brought me in starting as a child, working in all areas of the company, from quality control to media production to anaerobic chamber manufacturing. So I really grew up in an anaerobic environment. We always joke that I'm a facultative organism where I live in the air and also grew up in this anaerobic environment. But that's really what got me into it, and I've just been around it all my life.
That's great. Um, so, all right, it's my understanding that Anaerobe Systems offers what you call TruPRAS media. So can you explain what that means and also really, what makes that special compared to all the other culture media out there that's designed for anaerobes?
Sure. Well, PRAS to start off with stands for pre-reduced anaerobically sterilized culture media. And that's something my dad, when he started the company, patented and was the first person to come up with this process where everything from the time the media is heated up to sterilized until it's poured, packaged, and transported to the microbiologist, it's never exposed to oxygen. And that's really what the TruPRAS means. So during that sterilization process, if there's a great diagram showing, a high level version of how we make our media. But in that can there on the left, we sterilize the media. During that sterilization process, all of the oxygen is driven out of the liquid media while it's heated. And from that point on, it never touches oxygen. So the media is piped into large anaerobic chambers where it's filled into the plates or tubes, and it goes down conveyor belts where it dries and is stacked up and moved over to our packaging machine inside of these anaerobic chambers, and then packaged in aluminum foil pouches under nitrogen, never touching oxygen. Um, so no other culture media is really made this way. A lot of the culture media, most people, even for anaerobic bacteria, it's sterilized and poured in oxygen. It's stored in oxygen, and then it may need to be pre-reduced. Some other media, they might call it PRAS, where they still pour it in air and they just package it anaerobically, but it still has some exposure to oxygen. So the true PRAS media is not just pre-reduced anaerobically sterilized, but it's also just it has never been exposed or touched oxygen from the time it gets mixed and sterilized.
And why is it actually so important that media has never been exposed to oxygen? I would assume that, you could just put the media in an anaerobic environment a few hours before you're going to use it, and then it would become pre-reduced, right?
Yeah. And this is a perfect visual example right here of why that's the case and why it doesn't work. So I like to think of this culture media that you just autoclaved as a whole uncut apple. As soon as you pour that media into a bottle, tube, a petri plate in the presence of oxygen, it's going to start oxidizing just like this apple. You might not be able to see it in your culture media, but the oxygen is reacting with some of the nutrient components in there, the things that the organisms like. It's oxidizing and changing them chemically. Now, if you take that apple that you see here, you cut it open in the air and it turns brown, you put that back in an anaerobic environment or an anaerobic chamber, the brown doesn't go away. It's permanent oxidative reaction and damage to the media. Think of your media the same way as if it's made out in air and you put it into that anaerobic environment, there's permanent damage that's irreversible. The brown won't turn back. The media won't turn back. And I always like to stress that anaerobic bacteria aren't necessarily killed by oxygen itself, but they're really harmed by the oxidative byproducts and the reactive oxygen species, things like the hydroxyl radical, the superoxide anion, hydrogen peroxide. These are all things that are byproducts of the anaerobic metabolism from the organisms, but also oxygen reacting with components in the media. And those are the things that really affect these organisms. So preventing media exposure during the manufacturing and handling process, preventing the exposure to oxygen is really, really important to have later success when culturing with organisms. And I always like to go back to my dad's founding theory when he started making this media. Is very simple: is anaerobic bacteria don't like oxygen. So if they don't like oxygen, let's just try to remove oxygen from all aspects of handling culture media rather than trying to figure out a way to cheat the system and get a little bit of oxygen here and a little bit of oxygen there. If we eliminate oxygen altogether, we get very consistent results.
Hmm. Yeah, that makes a lot of sense. And I think this is such a great visual that I probably won't ever forget. So, you talked about if there's never been any oxygen in the media, you don't need to worry about those toxic byproducts that are coming from the oxidative reactions. And so, in this case, you can completely rule out that variable for experiments that you're doing. Are there other advantages that you get out of making media this way?
Oh yeah, definitely. And so this TruPRAS manufacturing process that really is unique to Biolog has some other great side effects, you could call it, and advantages of the way that we make it oxygen free. Because the media is pre-reduced with no oxygen absorbed into the media or the plastic or the glass that it's contained in, it can be inoculated immediately without a lengthy pre-reduction step.
Mm.
Most culture media that's stored in air or manufactured in air really should be brought into an anaerobic environment several hours or even overnight is best before you're going to streak organisms or inoculate organisms onto that. This allows the media to get into the reduced state. It allows all the oxygen to desorb or come out of the media. Even the plastic, the petri dishes, will absorb oxygen that will leach back into the media over time, even though it's in an anaerobic environment. So this TruPRAS media saves both time and space that you need to use for this pre-reduction step when preparing for anaerobic culture. The TruPRAS culture media also has a longer shelf life, so this is a great thing for planning on how often you need to purchase media. But it has a 3-month shelf life for agar plates and a 1-year shelf life for liquid broth, compared to most other agar plates that have a 6-week shelf life from the date of manufacture. And when you're making culture media yourself in the lab in a research setting, which oftentimes people do, really that should be used, within a few weeks of it being made. Um, and then the other thing is that since there's no oxygen in the packaging, and the media is not exposed to oxygen inside that package, it doesn't need refrigeration. So you can just leave it in the package in the box under a bench or in a cabinet. You don't need to use that valuable refrigerator space or buy extra refrigerators for storing all your media.
Hmm. Yeah, that makes sense. So now that we kind of know how the media is made, let's look at the different categories of TruPRAS media that we can sort of, divide up into these three main groups. So can we talk through these together? I think the first category is really for specimen collection and transport. So can you talk a little bit more about what these specific types of media are for and what they do?
Yeah, transport media really it's the first step in having success in anaerobic culture, isolation, identification. I always like to think of the transport media as it's the lifeboat that's going to get the organisms from wherever they're being collected, maybe a wound, an environmental site, maybe
Mm.
colleagues in another area. To safely get those organisms to a point where they can be cultured in an anaerobic environment. So our transport media comes in several different formats and sizes. There's some great examples in the pictures you see here. So we have surgery packs that are designed to go into the OR, operating rooms, or other sterile collection sites to collect specimens. So you'll see those in the peel apart paper packaging where they've been autoclaved ahead of time. And then we're the only manufacturer of a wide mouth anaerobic specimen collection designed for tissue biopsies, bone, or stool. So you see that there in that center picture where it's about a 25 millimeter or 1 inch diameter tube where you can take the top off and put larger specimens in. And really where it comes into play is when that specimen gets to you in the lab. It can be very hard using your tweezers to get those specimens out. So those wide mouth are great for larger biopsies or even stool or environmental samples. Um, Over there on the left side, you see a small shorty vial. We have a liquid 1 ml version of this transport media. This is used a lot for dental, like paper points for dental samples, but it works great in a research setting if you're thinking about collecting mouse pellets from a mouse lab and you want to move that across into your lab for workup where you can drop the pellet in there. And we've even had researchers had great success using that liquid transport media to directly use for a fecal microbiota transplant with that liquid sample into a mouse model, for example. Um, and so our standard tubes have a septum hungate cap on the top, so you can inject directly in. You can also remove the top if you need to, because we have special components in to counter the harmful effects of the media. And speaking of the chemistry and the composition of the transport media is really specifically designed to prevent samples from growing and metabolizing. That's the key step in transport and collection is that the metabolism and the presence of oxygen is really what harms the organisms. So we keep them in stasis. They're balanced and happy, but not growing for up to 48 hours before they get to the lab, and then they can be recovered and worked up in the lab.
Hmm. Okay. And it looks like the transport media that you've been talking about has really been widely recognized as, the best way out there to collect and transport specimens for doing clinical diagnostics of anaerobic infections.
Yeah. So this is a great example of Quest Diagnostics, really a leader out in the field, um, who has made a industry standard for all of their patient specimen collection and transport to them. Um, so they've updated their specimen submission guidelines to instruct that all anaerobic specimens be collected and transported in biologs transport media. So you'll see there, we've got the pictures of our wide mouth tissue transport and the standard Hungate cap transport. Just because of the success, the reliability, and the consistency you get using this transport media from collecting in surgical sites, surgery centers, clinics that are going to go back to a central lab.
Yeah, that's very important. Okay, so then the second category, I think anybody who's ever worked in any kind of microbiology lab is very familiar with agar plates. So these, there's got to be obviously something special about these plates. So can you talk through the plated agar media that's pretty highly enriched for these types of organisms?
Yeah, and we really make a wide variety of agar-plated media and formulations for clinical diagnostics. Also, general enrichment and growth, targeted microbiome organisms. We've
Mm.
got the selective and differential. So there's a wide variety that you use in different cases. So starting with that general enrichment, it's really your starting point for culture. And this is agar media that's designed to grow just about everything. So Brucella blood agar, that's industry standard for general enrichment, and it's supplemented with vitamin K and hemin and cysteine, which are really important for anything, any media where you're growing anaerobic bacteria. But we also make some more highly enriched plates for the more fastidious, which is organisms with really complex nutritional requirements. So we have culture media that contains things like short chain fatty acids, complex carbohydrates, sugars, serum, and complex vitamin mixtures. Some examples there are like our MTGE, the media that grows everything, with serum, YCFA, yeast calcitone fatty acid, with short chain fatty acids in it, and vitamins, and chocolate agar that has NAD. So those are some examples of highly enriched that maybe go beyond the general clinical diagnostics that are great for microbiome biobanking or just trying to grow everything that you could possibly want to grow.
Hmm.
Um, on the selective and differential medias, those are really important because they're a helpful tool in trying to isolate organisms from mixed samples. So this happens both in a research setting, for example, stool coming from microbiota sample or saliva, but in the clinical setting too. Most anaerobic infections or infections that you suspect an anaerobe is there have anywhere from 3 to 4 organisms that you're going to recover, but one of those is really the one that you're trying to target. So having this selective media can really be helpful in getting rid of that background noise or organisms that are not of interest. So in that top left there, you'll see a split plate with our BBE agar on the right, the one that's black. That's something that should always be used in clinical diagnostics because the BFRAG group is typically found in about 30% of anaerobic infections. And if it's B. fragilis or the BFRAG group, it's going to turn that media black, and you can give an early presumptive ID while you're doing further workup. Then on the left of that is our lake blood with kanamycin and vancomycin. We also have phenylethyl alcohol that's used that will inhibit the facultative organisms. So that really narrows you down to just the strict anaerobes that maybe you might be looking for, and you don't really care or don't want to try to identify or isolate those facultatives. Um,
Hmm.
and then we've got other selective media that can help both in diagnostics and on the research side. So we've got selective media for lactobacillus and bifidobacterium. In that top right, you'll see our CCFA for selective and differential for Clostridioides difficile. There's an egg yolk agar there that'll show lipase and lecithinase reactions. Um, but even if you're not doing clinical diagnostics, using these same selective media strategies and types of media can really help out in a research or a microbiome-based strategy for isolation to knock out a lot of the other flora when you have a highly diverse sample with maybe over 100 unique species, trying to narrow down to that one that you're trying to isolate.
Hmm.
And the one thing just to add is that we do make specialty convenience or combination packs, which are great on the clinical diagnostics side. Or if you just want one package, you can open a single pack, and it's going to have anywhere from 3 to 4 different media types in that one package. So as one sample comes in, one package will give you all the media you need for workup of a single sample.
Great, thanks. So the last category of media here is liquid media. This is also highly enriched. Can you talk a little bit more about, what these tubes or bottles of growth media are typically used for?
Yeah, sure. Liquid media is really similar formulations and categories as the other media we just talked about. It just doesn't have the agar or solidifying agent. So the
Mhm.
agar media is great for getting isolated colonies, and the liquid's best for when you're trying to get enumerations or growth promotion, things like that. So in a clinical lab, liquid media such as chopped meat broth or thioglycolate broth are used for initial screening of samples. So you have a sample that come in, and it might be from a surgery site where maybe it isn't, you don't expect an infection to be there. So that sample can go in a broth and observe it for 24 hours. And if you don't see any turbidity or growth, then you don't need to work it up on the agar plates. But if you do see turbidity or it looks like things are growing in that media, then you know that there's probably an anaerobic organism and you should work it up. On the
Mhm.
research setting, organisms, they might be grown up in broth to really build up the biomass to get more CFUs in there that you might be later inoculating into a mouse model or a larger bioreactor. And so with our various different broth medias, we have very consistent, good growth of these organisms to give you that success later. We make them in tubed versions. We also make them in large 250 mil, 500 mil bottles that can help for really getting large numbers of organisms to go into a bioreactor. And we also do, very similar. We have enrichment media. We have selective media, um, like lactobacillus broth. And we also have some very highly enriched versions of this, like our white CFA, yeast costitone fatty acid, MTGE, oral treponeme enrichment broth. Um, and then we have this specialty liquid broth for C. difficile, Clostridius difficile, that'll start out red, and you can inoculate it with a very, very small number of spores or environmental samples. And if C. diff is there, it'll inhibit all the other organisms. The C. diff will grow and it'll turn it yellow for a presumptive C. diff positive.
Cool. All right, so as we've gone through these different types of media, you've given some great examples of where they're used, in studying the microbiome or in clinical settings. Could we just talk about, are there any that we've missed here? Are there any main areas or fields where these media really are helpful, particularly, for some of these areas?
Yeah, yeah. So the TruPRAS culture media is really used widely in the diagnosis of infections where
Mhm.
an anaerobe is suspected, and that really happens both in a hospital setting with people, but also in veterinary and agricultural where you have animals that might be getting infections with their hoofs or looking at other things there. So that diagnostics isn't just just with people.
Mm.
Um, and then the multiple media types really help people to narrow down and identify the actual organism causing the infection in these situations.
Right.
um, the TruPRAS process and quality also leads to a lot of success in the research fields such as microbiome, biobanking, growth of these really fastidious, hard to grow organisms. So the highly enriched and oxygen reformulations give consistent results. So when you're repeating growth over and over for maybe a susceptibility panel or
Mhm.
just overall prepping inoculations going into a bioreactor, the consistency really is key here in all these areas.
Yeah, that's great. And I mean, it's so exciting to hear about things that are, very hard to culture or very fastidious that, you can get successfully growing because there's, I'm sure, just a wealth of knowledge that still remains to be learned. Actually, speaking of that, could we talk a little bit about some sort of, you know, um, I don't know if success stories is the right word, but just really some clear anecdotes or cases where it's so important to see, why you have to keep everything oxygen free in order to have very reproducible growth of some of these really tricky organisms?
Yeah, sure. One example is a research group that we were working with, doing microbiome research with young infant microbiome, and
Mhm.
they were using standard BHI broth and BHI agar for growing these organisms, which we make also. But making it themselves, they weren't using the TruPRAS method. They weren't using the same supplemented version that we were. And so we said, hey, why don't you try ours? I know you're using BHI, but try our media. And there were two organisms that they just could not consistently grow. And as soon as they used the TruPRAS BHI broth that we were making, it solved all their problems. They got great growth, consistent every single time. And we
Hmm.
found similar too with especially like the YCFA media and broth that we make. This is a really difficult to make media. It smells bad. It's hard to get the pH right. And it's not something that most people make commercially. So, making these complex media ourselves has really helped people get away from making it themselves and having inconsistent results,
Hmm.
out in the field. And one thing, this graphic that we have on the screen here, I always love to show this. It's a really great example when you talk about making it anaerobic, the TruPRAS method, the way we do it with the consistency and how oxygen really hurts here. So when we look at these are two different organisms that were inoculated into our MTG broth media with completely done anaerobically on the top, and you get consistent good growth across all 96 wells. And that bottom row there, all that was done is that media was brought out into air for a short period of time, about 30 minutes. The organisms were inoculated into that culture media, and then it was immediately put in an anaerobic chamber. So it was incubated anaerobically after that. And you can see the inconsistencies that you get. So sometimes it might grow, most of the times it might not. And then sometimes when it grows, it's not as good. So that variability is really what you get when you don't have good oxygen protection or avoidance of oxygen in your culture media.
Wow, yeah, that's really, I mean, it's also very organism dependent, obviously. So you just really want to make sure that you're keeping all those variables out out of the equation here. Um, yeah, that's very, very interesting. And you've shared a few tips and tricks already with, some of the stories you just shared about the switching media. Is there any other special tips and tricks to share with people who are working with anaerobes and maybe struggling, or um, any
Yeah, of
other?
course. One thing is always just starting with your transport media is just
Mhm.
making sure that you've got good specimen collection and transport. On the clinical side, one of the things is you always want to avoid swabs.
Mm.
These wounds and infections, they have multiple organisms, and it's hard to get down to where that anaerobe is with a swab. You'll pick up a lot of normal flora in the area. So using an aspirate or a tissue biopsy is really the best when you're getting things there. A couple other things that people always ask is, well, how long can I handle my organisms out in the air? And obviously the best is using an anaerobic chamber. You do all your culture inside, but if you have your pre-reduced media, this TruPRAS media, it can be opened out in the air, and you have about 15 minutes really before there's any oxidative damage to get that into an anaerobic environment. So you don't
Uh-huh.
have to get it in there immediately, but some exposure out in air. But you really want to get, and once that organism touches the culture media, it's going to start to metabolize. And if it's sitting in oxygen, that's really what you want to avoid. Um,
Hmm.
and then the last thing is just having a strategy for the right media. Oftentimes you need multiple media types. One media is not going to just be the silver bullet and grow everything. So you may need some selective media. You may need different types of enriched media to get that organism isolated and cultured and identified.
Hmm. That's really helpful. Thanks. So finally, I'll just remind everybody that we've sort of talked about the three main categories of media, but you can find the full, pretty extensive list of all of the different media products that we offer at Anaerobe Systems, which is now part of Biolog, on our website, which is listed here, or you can reach out to us for more information about any of them. And we're really excited to now offer these products outside of North America, and that's either directly or via our network of distributors. So please go and check our website to see how best to purchase them, or just send us an email at info@biolog.com.